Chapter 10, Section 2 discusses the structure of DNA, or deoxyribonucleic acid. It is an organic compound, which means that it is carbon-based, and it's made of repeating subunits called nucleotides. So we talked earlier in the year about how DNA is a polymer or large molecule, and its monomers are nucleotides. And it's made of two long chains in this double helix structure. Sometimes it's also referred to as a twisted ladder. So it looks kind of like a spiral staircase. So each nucleotide is made up of three parts: the sugar molecule, which is this central portion down here. In DNA, it is deoxyribose. Attached to one side of that sugar is a phosphate group, and attached to the other side is a nitrogen-containing base. This nitrogen-containing base can be one of four different bases, so it allows for some more variety. These are the four bases: adenine, guanine, cytosine, and thymine, and most of the time we just refer to them by their first letter, so A, G, C, and T. Now these bases are divided into two categories: purines, which are double rings of nitrogens and carbons, and pyrimidines, which are single rings. Now when these bases are paired up in the actual DNA molecule, you always pair a purine with a pyrimidine. So you end up having these three structures all attached to each other evenly, instead of two being paired with two and one being paired with one. An easy way to help you remember which ones are purines and which ones are pyrimidines, the way I do it: cytosine has a Y in it, thymine has a Y in it, and so does pyrimidine. So that's how I remember that those two are the pyrimidines, and then that must mean that adenine and guanine are the purines. The double helix structure of the DNA molecule is mainly credited to Watson and Crick. Back in 1953, they were the ones that suggested the model, and they finally really were able to break through and figure out this structure with the help of Rosalind Franklin and Maurice Wilkins, who did research with X-ray crystallography. This is one of their pictures here, and that's what finally kind of gave Watson and Crick that edge to finish off their diagram of what they believed the DNA structure was actually looking like. In the DNA molecule, we refer to it kind of in two parts: the backbone and the rungs of the ladder. The backbone is made up of the sugars and phosphates. So here's one nucleotide, here's another nucleotide. They're all bonded together. This is only one strand of the DNA. You would also have a second strand here. But these sugars and phosphates are covalently bonded to each other, so they're fairly strongly bonded to one another. The bases are going to be the rungs of the ladder, so they're perpendicular to this backbone. Now the bases are going to be held together by hydrogen bonds, which we talked about early in the year. Hydrogen bonded to something more negative than it, generally nitrogens, oxygens, and fluorines. So we know we have nitrogens in these bases, so that's going to help to form those bonds. And we need these easier to break, weaker hydrogen bonds holding our bases together so that we can replicate this DNA. But we want the stronger covalent bonds in the backbone so that we don't lose the whole structure of our DNA and have this part break. And finally, there are some rules which are referred to as complementary base pairing. Sometimes they're referred to as Chargaff's rule, after a gentleman named Erwin Chargaff. And what he figured out was that percentage-wise in a DNA molecule, C and G generally have about the same percent, and so do A and T. So the rules are that C, or cytosine, always pairs with G, or guanine, and adenine always pairs with thymine, or A with T. So the nucleotide sequence of one chain is considered to be the complement of the other chain. So if I have A on this side, then I automatically know that on the other side of the strand will be T. If I have a G here, that means on the other side it's going to be a C. So here's those rungs of the ladder with the hydrogen bonds attaching our bases, and then here's the backbones that are covalently bonded with our sugars and phosphates.